In this video, I want to go through the proper way to write a Punnett square and to then analyze a Punnett square after they've been drawn. This will probably be a review for a lot of you, but that's okay. I just want to make sure that we're all on the same page. So before we start, I just really want to stress that what we're looking at here, whenever we're talking about alleles and genes, are sections of DNA that are coding for proteins that give us our traits. So whenever you're writing these letters, in this case, I'm using uppercase F for the dominant allele and lowercase f for the recessive allele. But whenever you're writing these letters, remember that you are just labeling genes. This is an actual section of DNA that you are analyzing. So here we would see the maternal chromosome for chromosome 1. It has the recessive allele, and the paternal has the dominant allele. Here I'm using the letter F. We'll be looking at how freckles are inherited. Freckles, those dots on your skin, are thought to be controlled by mostly dominant genes, but with genetics, everything gets kind of muddled, so it's hard to say for sure. But that's the example we're going to be using. So the dominant allele F will be an individual that does have freckles, and the recessive phenotype would be lowercase f, someone who does not have freckles. All right, whenever you're drawing a Punnett square, this first one that we're going to do is only looking at one gene, freckles. You need to just draw what kind of looks like a window pane. So it's just going to be a box. And inside of the box, you'll draw a line that divides it into left and right and then up and down portions. It kind of looks like a tic-tac-toe board. And this is going to set up our Punnett square. It's also helpful to write out the genotypes of the two individuals that you are going to be doing a cross of. What you're basically doing is you're looking and seeing, okay, what would happen if I took this individual who has both copies of the dominant allele and crossed it with this individual who has one copy of the dominant and one copy of the recessive, so a heterozygous individual. In order to get this originally set up, what you need to do is figure out all of the different possible combinations for each of the parents and list one of the parents' genotypes across the top and one across the side. It's easy when there's only one trait because there's only one possibility. So across the top, I'll put this individual who has one big F and another big F. And on down the side, I'll put the other individual who has one big F and one lowercase f. All right, now you're ready to actually fill in the Punnett square itself. In order to do this, I call it battle shipping. You're going to kind of treat this like a grid where you're going to bring across the allele and then down the allele for that box. So we come across, this will be big F and big F. And then in this same box, we'll come across and down. So big F, big F. Then in this bottom box, we'll come across, lowercase, and down. I always put, if there's going to be a dominant allele, I always put it first, like that. And then same thing here. So that would complete our Punnett square. Once you have your Punnett square complete, then you can go and analyze the different phenotypes. So this individual would have the dominant trait because he has two copies of the dominant allele. Here we see another one that would have the dominant trait. Here we would see one that also has the dominant trait because even though it has one copy of the recessive gene, it would take two copies in order for you to see the recessive trait. So this would be dominant as well. And here we would see another dominant individual.